Hey, hello everyone. Today we're going to talk about this spermatogenesis versus oogenesis. Yes, बहुत important topic है. And see, while differentiation, we are going to cover some points of the spermatogenesis, some points of oogenesis. That going to help you to understand the concept. And definitely, my next video will be on what is exactly spermatogenesis, what are the detailed studies, and what are the oogenesis. So let's start with great enthusiasm and let's study with Sumit Madik with a fantastic way. So here we are starting the things. So now here we're going to talk about the spermatogenesis and oogenesis in point-wise manner. So it will be helpful to you. First of all, site of occurrence. Where does spermatogenesis occur? Obviously in seminiferous tubule of the testes. Seminiferous tubules consist of leading cell, spermatogonia. Yes, right. So these are the consistence of the seminiferous tubule. Okay, and this is spermatogenesis is the site of which is the site of spermatogenesis? A seminiferous tubule will be there, and leading cells is also there. Okay, now oogenesis obviously in going to occur in a ovaries definitely. Now total period, how much time you take for the spermatogenesis and oogenesis? It is continuous process. The sperm production in the male is continuous. It's never stopped. So it is completed within 74 days in the humans. Okay, so for the spermatogenesis, from the spermatogonia, if the spermatids are forming and it's undergo the maturation, it takes around 74 days. Where in case of oogenesis, it's completed in minimum 12 to 15 years. Can you understand what is the big difference here? Years. So if the female is there, definitely when she is just born, she birth with a ovas. Okay, and that antrum or we can say that cell is continuously giving the eggs. That is menarche, from the menarche till the menopause. So this is a period, a reproductive period for the female where she is going to give the yes oogenesis things. Okay, so it's a 12 to 15 years growth phase of the shorter duration. The growth phase of the spermatogenesis is what? A very shorter. But as compared to oogenesis, it's a longer duration. Oogenesis growth phase is longer, definitely, as you can see here. And here, in case of spermatogenesis, is a shorter one. Yolk synthesis. Okay, no yolk synthesis in this phase. There is no yolk. In oogenesis, what is actually formed? The egg. So ova covering should be there. Yes or no? Covering should be there. अंडा देखा है आपने, राइट? येलो कलर का बीच में होता है और अल्ब्यूमिन होता है उसका आजू बाजू. सिमिलर वे ये क्या है? The primary membrane which is forming, see this is actually egg, and the primary membrane which is forming outside, it's called a vitellogenesis. In sperm, there is no need of the formation of that because sperm has motile and they has to be transferred for the fertilization purpose. But in case of egg, it has to be fertilized, so it has to be protected. So vital genesis is a process in which a primary layer is going to form. So it is only occur in oogenesis. Now nuclear changes. What are the nuclear changes over here? Nucleus become condensed by the loss of superfluous material. Nucleus is condensed. Nucleus is bloated due to the increase in nucleoplasm. So in oogenesis, nucleoplasm is much more as compared to spermatogenesis. So these are superfluous material is present in where? Spermatogenesis. And the nucleoplasm is present where? In oogenesis. This point is very clear to you. Yes, so please be aware of that. Then we're going to talk about the number of gametes. One spermatogonium. Can you see that? This is a spermatogonium. It undergo first mitosis, then two meiosis will be there, and then we are getting four what? Sperms. Which sperms are haploid, correct? So at the end of the spermatogenesis, from the one spermagonia, we are getting four sperms. How many? Four sperms we are getting. So this is a product of the spermatogenesis. But in case of oogenesis, we only want one ova, one egg. Definitely we want one. So one oogenesis undergo synthesis changes, one mitosis again, and two meiosis, and two polar bodies are formed. That is removed off, and one egg, which is haploid ovum, is we got. So this point is very much clear to you. Now polar bodies. Obviously, in case of spermatogenesis, no polar body is going to form. What is polar body? See, we want to make to haploid. क्या बनाना है हमें? Haploid बनाना. अब अगर haploid बनाना ही है, तो we are using that 4 cells. But in case of oogenesis, we want only one egg cell should be there. So for that purpose, polar bodies which is form of the meiosis 1 and meiosis 2, that has to be removed off. So how many polar bodies are there? 2, or maybe sometimes 3 is also there. This point is very clear to you. Yes. Now let's move to the next point, that is site of completion, where actually spermatogenesis actually get completed. It is started and completed within the testis only. But in case of oogenesis, it started inside the ovary, primary, secondary, tertiary, graphene follicle. Then that graphene follicle undergo mature and the ova in the influence of FSH and LH. Okay, but it generally completed outside the ovary into the oviduct. What is oviduct? Fallopian tube, isthmus, in ampullum, and infundibulum, right? That is the things over there. The fallopian tube is there for oviduct. So where the oogenesis is actually done, completed in a fallopian tube, and testis in which spermatogenesis is completed. So these points are clear to you. Let's move to the next points here. Now, the moment I say polar bodies. So not form here. There are only 2, 3 are forming. Why there is no polar body in spermatogenesis? You know the answer right now, right? Now, site of completion that we already talked about. The last one, size of gamete form. So sperm is much smaller than the spermatogonium. See, spermatogonium is a very big cell, but when it's undergo the mitotic and two meiosis, they are going to form like this. Okay, the small one. So size of the spermatids actually undergo the maturation. Spermiation is done, tail is there, and they undergo maturation in a beta differential. Okay, what going to happen in that case? They undergo maturation, and the sperm is smaller than the spermatogonium. But in case ovum is much larger than oogonium. Oogonium cell was very small. Can you see that? Okay, and ovum is going to form a larger one because they undergo the series of the changes because the primary layer should be there. Vitellogenesis is there, right? If you have just studied that, how can you forget, my dear students? So let's go and just again revise what we want to talk. So in this case, okay, in this case, we talk about what is that we talk about? Hmm? See, vitellogenesis occurs in a growth phase. So they come इन केस ऑफ स्पर्मेटोजेनेसिस क्या चाहिए हमें उसको रिड्यूस करना है हैप्लॉइड करते करते तो एक बड़ा सा सेल लेगा उसको 4 में डिवाइड करना है दैट्स इट बट इन केस ऑफ ओवा दैट इज क्वाइट डिफरेंट छोटा सा रहेगा उसको बड़ा बड़ा करते रहना है बिकॉज layers बनते जाएंगे so that is a actually difference between the spermatogenesis and the oogenesis. In next lecture I'm going to talk about spermatogenesis in detail, what are the steps and all, and the oogenesis I'm going to talk about in detail. So are you ready for that? Everybody is ready for that, right? So let's meet in a next class regarding spermatogenesis and oogenesis. Different lecture I'm going to make. And don't forget that I'm just one comment away from you. If you want any video, just let me know this topic I want. I definitely try my best to give you that output. So thank you so much. I hope you enjoy the session.